Our next speaker is also a recipient of an Ann Miller Gonzalez grant for graduate research. Silas Jenkins is here with us today. She's a native Austinite and grew up here, and her parents are here today listening to her, so we're excited about that. They're members of the Austin chapter. And Silas is a grad student in the biology department at Texas State University in San Marcos. Their research focuses on the potential effects of inoculum source and mycorrhizal status for Quercus buckleyi, also known as Texas red oak or Spanish oak. And so that effect and drought stress response at the seedling stage. The greenhouse study will endeavor to answer the overarching question: does mycorrhizal inoculum affect drought response in Quercus buckleyi seedlings? So I think these two ladies need to get together and have a conversation after this. We're really excited to hear about your research, Silas. Thank you.
Hi, y'all. So I'm also, this is a work in progress. I'm at the sort of ready to share some preliminary results. Oh, right, the microphone. Okay, so my thesis experiment involves the mycorrhizal relationships of, well, you said it already, but before I get too deep into the questions and the design, I want to just explain for those who might not know what are mycorrhizae. So it comes from the roots myco, fungus, rhizae, root, the ubiquitous mutualistic symbiosis between pretty much all plants, all vascular plants, and certain taxa of fungi through the plant roots. And so the simplest explanation, right, is that the plant gives carbon from photosynthesis to the fungi, and the fungi give, well, they confer all kinds of benefits to the plant nutritionally. Sometimes they provide the plant with all of their macronutrients, micronutrients. They also help protect from pathogens and toxins, and they've been shown to ameliorate plant water stress. And there's a variety of mechanisms by which that can happen. But there are a few different main categories of mycorrhizae. The more ancient monophyletic lineage is called arbuscular mycorrhizae. They're called that because they form these little tree-looking structures inside the root cortical cells. So they actually penetrate the root cortical cells, and they don't form fruiting bodies. They're obligate symbionts. That's more than 70% of plants form these arbuscular mycorrhizal relationships, AN. And then there are certain plant families like orchids and heath family plants that have their own mycorrhizae. And about 2% of plants, mostly these are forest trees, pine are one, oak are one, they form what are called ectomycorrhizae. And so ectomycorrhizae, they're harder to generalize about. They independently evolved at least 60 times that we know of. So they're not a monophyletic lineage. They evolve from different ways of life. And they're called ectos because they don't penetrate the root cells. They form a hyphal mantle or sheath around the root tips. And oaks are special in that they can do both AM and EM. That's not super common. And studies have shown that they can benefit nutritionally and water stress wise from both types of relationships. So my questions, I looked at whether if a seedling were provisioned with soil from different ecological site types, does that affect what partnerships they form? And do those partnerships influence how they respond to drought stress? Like looking at growth and photosynthetic rate in particular. So I hypothesized that it will influence, where they get their inocula from will impact which partnerships they form, and that my guess was just that under well-watered conditions, they would have the most root colonization, like more of the root tips would have mycorrhizae, and more diverse mycorrhizae the closer they were to home soil. So if they got soil from beneath a mature oak, they would have the most colonization, the most diversity, probably grow larger and have higher photosynthetic rates. And I thought that there was a chance that under a drought scenario, they could actually benefit more from juniper soil inoculum, because juniper are arbuscular, and arbuscular mycorrhizae are thought to tolerate temperature and moisture extremes more. So I thought they could have more beneficial mutualists there. Then there was a inoculation treatment from clearing soil. So the further you removed you are from the forest, the less likely you are to have ectos. I mean, studies have shown that. And so I thought, they might not have many beneficial mutualists with clearing soil. So I thought they'd be smaller and have lower photosynthetic rates. So those were my best guesses going into this. I conducted a factorial randomized split block design greenhouse experiment. So there were 4 different inoculation treatments and 2 water regimes, so 8 treatment combinations, spaced across 13 blocks. So one half of the block was watered, one half of the block was droughted. In each half, there are 4 reps. It's a total of there were 52 reps, 416 seedlings. Started by collecting acorns from their home, from from Riker Ranch, which is a tract on the BCP. And I weighed all of them. And I did this because there have been shown to be an inverse correlation between mycorrhizal dependence and seed size. So like a smaller seed would benefit more from mycorrhizae, and also, they have more cotyledon reserves if they're bigger, and that would impact their size. So I weighed all of them, seeded all of them, seeded over 1,000 of them. So I had a failure the year before, but anyway, yeah, so I seeded a lot, sowed them immediately. That was the failure, actually. I stored them for too long. So sow your acorns immediately, especially with red oaks. But anyway, so they emerged. I documented their emergence, and in March, selected participants who were all more or less the same size. And when we transplanted them, that's when we added the inoculum. And the inocula came from 2 different locations on the Balcones Canyonlands Preserve. This is a map of the sites at Riker Ranch. This is a older forest. The blue dots are where the oak trees were. These are in a closed canopy juniper oak forest. These are all Quercus buckleyi conspecifics. And then the red triangles are where there were mature junipers, but removed from conspecifics. And so those were 2 inocula, O and J. And then 2 inocula came. And so here's just so you get a sense of scale. We were going for trees that had basal circumferences of at least around 100 centimeters. So like larger elder trees. And that's because the larger elder trees have been shown to have more mycorrhizal connections. So then Cortana has a different land use history. It's also on the Balcones Canyonlands Preserve, and it was cleared 20 years ago. And there are no Quercus buckleyi out there. There are no red oaks. There are Quercus fusiformis. So the clumps that you see are just mott's, woody veg of Quercus fusiformis and ash juniper and some other woody veg. And then, so we got what I'm calling the shade inocula from there, and within a meter of both a oak and a juniper. And then the clearing inocula minimally removed by 5 meters from any woody veg, even like a juniper sapling, but further from oak. So those are the 4 inoculation treatments. We transplanted them, and yeah, and here's a visual for what those sites looked like. And here's a cartoon of, this would be 1 block. So imagine the blue is watered, the red is droughted. There's 4 reps. We allowed them to kind of do their thing, establish connections, and recover from transplant shock for about 7 weeks. And at the end of May, I ceased to water the drought treatment. They did not get any water for the duration of the experiment. Whereas the well-watered group, they got watered to field capacity once every week. At the beginning of June, I started collecting gas exchange measurements with this little contraption that's like a Lycor, but it's a CRAS. Anyway, and so I was looking to see, do they respond differently? Obviously, we imagine that photosynthetic rates are going to plummet with the drought. And also, I should say that our evaporative cooler was broken in the greenhouse and it was extremely hot in there. Like it made outside feel cool this summer. So they were all kind of stressed. Like this was a high stress situation for everybody. So let's see. Here's just a little, this is stomatal conductance. And I didn't find, well, I've analyzed it a few different ways, and this is still preliminary. But they didn't really start to feel the drought until 4 weeks in, which is really impressive. Like the well-watered and droughted groups didn't separate until 4 weeks in. And then they did, and there didn't seem to be significant differences based on inoculation treatment. However, this graph got a little wonky when I added the crispy leaves, but this is my time to crispiness graph. I conducted a log rank analysis to assess whether inocula was significant in how long it took them to get crispy. And by crispy, I mean, I think you know what I mean by crispy, but just kind of, yeah. So we collected a qualitative data on that, like, do they feel crispy? Yeah, they feel crispy. So on the x-axis, we have days from last water. This got cut off, but it's a single line until you get past 30, right? And so, and then on the y-axis, we have the proportion that are not crispy. So they're doing good. They're all at 1 until you get past 30 days, and then they start to separate. And so the orange line is the juniper inocula, the yellow is the shade, the blue is the oak, and the purple is the clearing. So here you can see that the biggest difference is between the juniper and the clearing. Like the juniper inoculated seedlings actually could withstand the drought a little longer, a little better, and then the clearing inoculated seedlings sort of succumbed sooner. We did a rolling harvest because I wanted to be able to preserve the mycorrhizae for microscopy. Anyway, long story short, we did a rolling harvest starting in the beginning of July that culminated on July 25th. So like about 60 days since last water. And by then they were pretty all senesced. And we preserved the leaf where we scanned the leaves for leaf area and then dried them and weighed them and dried the stems. So I have data on leaf area and leaf biomass and stem dried biomass. And the p-values that are significant are highlighted in pink. So no surprise that, the drought treatment influenced the size, right? That's pretty obvious. Also, the seed mass you can see is like highly significant for everything I measured. Seed mass was very significant. Block also was significant. And I have a couple ideas about why that might be. But as far as inoculation treatment, the only size measurement that was impacted by inoculation treatment was stem mass. So stem mass was the clearing seedlings got the biggest and closely followed by the juniper seedlings. But the significant difference was really between clearing and shade and oak. They were smaller. And the biggest difference was in the well-watered group. But that's all I got as far as size differences. It's a big sample size, so it's kind of interesting that there were differences. But then I washed the root systems, subsampled the root systems, and shipped them on dry ice to Northern Arizona University in Flagstaff, where they trained me in mycorrhizal microscopy techniques. Basically, what I was doing was looking at the root sample on a dish that has a grid and tracing the grid, and anywhere that the root tip intersected with the grid, I'm counting it, and I'm categorizing it as either colonized and living, colonized and dead, uncolonized and living, uncolonized and dead. And this is for ectos, by the way. Ectos are visible under a light microscope. And then also as a proxy for who they are until DNA sequencing can happen on more of them, I was morphotyping them. So I ended up creating basically 24 categories based on how they appear to me and keeping track of that too for community composition and richness information. So that's me counting them. And the biggest surprise, well, there are a couple of big surprises. One, so for arbuscular mycorrhizae, you can't just see them. You have to do this clearing and staining process. So roots, we cleared them with KOH and stained them with trypan blue. And this is not an image I took, but the blue circles are vesicles. You're looking for the hyphae, the vesicles, and the arbuscules of the arbuscular, and they weren't there. So that was one interesting thing to me because AM are more prevalent and oak are thought to be dual, and no studies have been conducted on this particular oak, but and also because AM are more resilient to or tolerant of extremes, I thought that they would have some, but they didn't in none of the treatments. Then they all had a lot of ectos, and regardless of treatment, and they were in the well-water groups 70 to 90% colonized, which is pretty high. And then in the droughted groups, you could tell that they had been colonized, but their root tips were dying because they were, hadn't gotten water in 2 months and were in pots in a hot greenhouse. So these were some that we were able to sequence that are pretty darling. This is a tuber, so same genus as a truffle. I called it cattail tip. I had names and numbers trying to keep track of all my categories, and then these are all in the same order, the Pizizales order, but different family. This is a Pyronymataceae, and then Piziza. But those are just a few, and there's a lot more that I still don't know who they are. And but it's been really fascinating. So the community composition work is ongoing. And yeah, I am grateful for the NEPSAT funding, and this work really wouldn't have been possible if not for all the mutualisms in which I am involved. And and a lot of folks helped with this project, and especially my mentor, Lisa O'Donnell, who's a senior biologist with the city of Austin. She kind of got me into this topic, made the connection with Lisa Markovchik at NAU, and then Kitty Gehring and her labs, Juji Kovacs. They were all just like, they taught me this and helped me learn how to do everything. And yeah, it's an ongoing collaboration, and I don't know, stay tuned, there will be more, but yeah, like,
You had said that the juniper was the best, that those microbes that you pulled from under the junipers were the best in drought conditions. Correct?
all I can really say is that they withstood crispiness the longest. And they also were oh yeah. The question was, it seemed like the juniper were the best for drought conditions. Yeah. Okay, sorry, I forgot to look at you entirely the whole time. I forgot. Okay.
I'm just thrilled. So the question from the audience is, would the soil under a juniper assist plants, or is the fungi specific to the oak or juniper? And I can repeat that.
I'm not sure I understand the question.
So is it, would the soil under the juniper assist so help plants, or is the fungi specific to oak or juniper?
Oh, I can see the question. Yeah, we don't know much about whether oak and juniper share symbionts yet. But my guess is that they might. But also, as far as we know, well, I could ramble about this. There's one study that came out of central Mexico a couple years ago, well, 22, that there are people who suspect that juniper might associate with EM, but that has never been shown. So, as I said, like, we didn't find any AM on any of my seedlings. There were a number of things I meant to say and forgot to say. One of them was that actually we did find a lot of dark septate endophytes. So if that means anything to anyone, that's kind of curious. And so I might end up analyzing that data too. Am I supposed to field questions? How does this work? Oh, okay, no more questions. Okay.
No, one more.
Oh, one more question. Okay. Oh. Okay, I don't know.
Okay, hey, so I'm just curious, there's a lot of business surrounding the association of mycorrhizal and juniper. especially in the duck that they create, it's kind of like a horticultural curiosity. But I'm curious if you actually were able to identify individual species that were present in the juniper duck, that substrate you used, that weren't present in the other soil techniques.
Yeah, I did not,
What was the
Summarize from the front quotes.
oh, sorry. I was, juniper duff is a horticultural curiosity. I don't remember what all this person said, but the main question was, did I look to see who was there in the juniper duff? And I did not. This was like kind of a potting frenzy, and I was trying to get the inocula, keep it for as short a time as possible before doing the transplanting, and also didn't have like, also, well, I mean, this is maybe a question more for the previous presenter, but as far as like metabarcoding for soil stuff, that gets really, well, expensive, but also tricky. And I think that some fungi get more amplified than others. There's also a lot that we don't even know who they are. Like some of the sequences I ran, you know, they haven't been registered yet. They're not on the, they're not known. And so, but the short of answer is no, I didn't unfortunately take that, but I could. That's future. I mean, that would be interesting. I'd like to know. Yeah. I was more interested in who's actually on the oak roots, who actually ends up, yeah, on there. Yeah. So thank y'all.
Okay, thank you to both our presenters. That was so fascinating. I would need another 10 years to probably understand most of that. But I think that interesting thing is that this level of research is going on, because I think it will, as we work to bring more Texas native plants to the nursery trade, I think this kind of research is going to be helpful in determining viable species in the marketplace. So I think they'll both find employment pretty easily, I'm pretty sure. So